Now see, I am explaining you all the step. Okay? Till you sew your hand gloves, I am explaining you from the first to the last. First, sample selection. Second, sample homogenization. Third is cell lysis. Fourth step is protein precipitation. After protein precipitation, we centrifuge it. In centrifugation, you see three layers. Top layer containing DNA. Middle one is impurities. Bottom one is protein precipitation solution. Clear? In DNA isolation buffer solution, there are four chemicals. First is trace, that maintain pH. Second is EDTA, that bind Ca++ Mg++ ions, stop DNase enzyme. Third is NaCl, that maintain ionic strength and help in DNA precipitation. Fourth chemical is option 2. If you add SDS, your kit will be ready for animal DNA isolation. Hair, blood, saliva, heart, liver, kidney, mosquito, housefly, that's. If you add C-type chemical, your kit will be ready for the plant world, fungal world DNA experiment. Clear? So, 5 chemicals in first DNA isolation buffer solution. Second chemical is protein precipitation solution, that containing phenol, chloroform, isoamyl alcohol. Phenol is too much smelling, so we avoid it, only use chloroform isoamyl alcohol that precipitate the protein by mixing in a different angle, turning it, right? After that you have to centrifuge. What they did in blood DNA samples, they take the blood sample, added RBC lysis and shake it. They centrifuge, cell get bottom attached, remove the liquid. They get WBC. Add WBC by pipetting, just did pinch it, in and out, liquid 10 times, broken down. Then add protein precipitation solution, same you added in the plant saliva and hair, and they mix it. Once they mix it, 3 layer is divided, same as your sample. Top layer they separated and added alcohol, good quality of DNA they see now, there. After that you will centrifuge again, like WBC again, DNA stick on the bottom, remove the liquid, finally you get the DNA. That is the process. After lunch, what you do? You transfer the top layer liquid, add alcohol and see the DNA. Electrophoresis, there is Agarose Gel Electrophoresis. Agarose powder, buffer solution, mix and boil. It cool and become the gel. There we place the combs, so they make wells. In each wells, a different person loaded the sample. A different sample you can load in each well. Okay, we apply the current over there, that will run the electrophoresis. So electric, 4 means holes, capillary size holes, and phoresis means movement. Under electrophoresis, movement of a molecule through the gel, so it's called Agarose Gel Electrophoresis. Another electrophoresis is used in biotechnology, that is SDS page. It's called SDS, polyacrylamide gel electrophoresis. Polyacrylamide gel is quite clear to your lens type. It also used to prepare the lenses, eye lenses. You know eye lenses? It is a water containing absorbing liquid. If you put on the eyes, it absorbs water and become clear, transparent, more than glass. Okay? So that electrophoresis is used to separate the DNA. That phase electrophoresis can also be used to sequence the DNA molecule. Clear? That is electrophoresis. What is left in DNA fingerprinting? Suppose what you do if you do the DNA fingerprinting. First you isolate the DNA, right? Then you do the electrophoresis to see DNA is coming out or not and what is quality of it. Once the electrophoresis is done, you dilute your DNA around, you dilute the DNA. Once the DNA is diluted, then 2 microliter from that diluted DNA you will take for one time of DNA fingerprinting experiment. Then what you start? You make the PCR cocktail, 5 chemicals together to make the DNA fingerprinting reaction. First is buffer solution. Second is D and TP mix, जिससे DNA बनता है ATGC. उसका मिक्सचर ATGC 4 आप ऐड कर देंगे. 2 चीजें ऐड होगी, बफर ऐड हो गया, DNTP ऐड हो गया. अब इसके बाद ऐड करेंगे प्राइमर, जिस जगह से कॉपी डीएनए की निकालनी है, टेस्ट करनी है, द पर्टिकुलर स्पेसिफिक सीक्वेंस, उसका अपोजिट सीक्वेंस 10 to 20 न्यूक्लियोटाइड का जिला में हम सिंथेसिस करते हैं, उसको डालेंगे, प्लस आपका डीएनए जो टेस्ट करना है, प्लस एक एंजाइम जो इस पूरी रिएक्शंस को कंप्लीट करता है, राइट, टैग पॉलीमरेज का, सबको मिक्स करेंगे। कितनी क्वांटिटी में? 16.5 माइक्रोलीटर वाटर, 2.5 माइक्रोलीटर बफर, 2 माइक्रोलीटर DNDP मिक्स, 1 माइक्रोलीटर फॉरवर्ड प्राइमर, 1 माइक्रोलीटर रिवर्स प्राइमर, so 2 माइक्रोलीटर प्राइमर, then 1 unit per reaction enzyme. Enzymes come in unit. इंसुलिन का एंजाइम आता है यूनिट में, एंजाइम्स कम इन यूनिट, so 1 unit per reaction, 1 unit enzyme, and then 2 microliter of your enzyme. Clear? That will put in the one tube, and tube will be similar like this tube we use, but they will be smaller size, right? And put on the machine. That's called PCR machine. और बटन प्रोग्राम सेट करके दबा वो कॉपी बनाएगा, और फिर आपको जेल इलेक्ट्रोफोरेसिस करनी है, उसकी फोटो डीएनए फिंगरप्रिंटिंग इज डन. नाउ स्टार्ट द एनालिसिस वर्क. In that you find out genes, allele, pattern, profile, diagnostic marker, everything. Once you return back, we will do the PPTs or part will be discussed. So you will be the scientist of molecular biology. Only 3-4 times if you repeat, you will be best biotechnology student.
Say thank you at least.
Understand? Raise your hand gloves and discard. यहाँ से पकड़ेंगे बच्चों, उंगली से नहीं खींचना है, यहाँ से पकड़ेंगे, उल्टा करते हुए इसको खींचेंगे और डस्टबिन में दुश्मन की तरह फेंकेंगे.